Next part of the program is going to feature Dr. Gil Levin. And Dr. Levin is going to talk about the oxidant on Mars. And I'm going to turn the program over to you right now. That's fresh water.
Thank you, John. Well, I see I have to follow a very exuberant performance, which in turn was followed by the real McKay. I can only hope to inject a little living into it. And that's really what I've come to talk about. The Viking 1976 missions to Mars were billed by the National Academy of Science as perhaps the most important scientific experiment in history. That is, to search for life on another planet. I think it was very interesting that in the introductory films prepared by NASA, you did not hear the word life. What happened to that $1 billion search for life? Was the evidence so conclusively negative that it's not worth talking about anymore? Or was there some reason to denigrate the principal purpose of the mission to, as it appears in the new book, Mars, a book of some 1,500 pages, to confine the chapter on the search for life to 11 pages. And in that text, to merely say, there are some scientists who believe that Viking detected life on Mars, but not to present their case. The truth is that for all of its promise, and despite the fact that it was probably the most complex space mission of our time, Viking did not solve the riddle and leaves us, as its legacy, two very important scientific problems. Did Viking discover life or some oxidant on the surface of Mars is the key problem. But another problem, really equally as important, is where are the organic compounds that everyone knew was going to be on Mars and constituted the reason to send a very sophisticated machine to determine which organic compounds were there. Not whether there were organic compounds there, we all knew there were, but which organic compounds were there. Strangely enough, the two questions remain. The Viking carried on each of its two spacecraft the contingent of 3 experiments to look for life, plus an experiment to identify the organic matter in the soil. I was experimenter on one of those experiments I would like to talk about, the so-called labeled release experiment. Originally, I called it Gulliver, but that didn't fly well with the bureaucrats. They did not see the analogy of going to a strange place to look for strange organisms, so they gave it the much more erudite name of the labeled release experiment, which we've contracted to the LR experiment. The rationale of the LR experiment was we could use the very, very sensitive method of radioisotopes to look for life. And the life we're looking for are not the green-eyed monsters, but tiny single-celled microorganisms, which we felt that were there any life on Mars, there would have to be microorganisms, because even if there were elephants, which would probably be hard to catch, they would require microorganisms to decompose them and keep the life cycle going. And if microorganisms on Mars had pursued a path of those on Earth, they would be distributed as a thin film throughout the entire surface of the planet. So this was the place to start looking for life. And we selected the most sensitive tool that we could. It was a tool that supplied radioactive carbon compounds to portions of soil, and were there anything alive in that soil that could utilize the organic compounds, hopefully, these organisms would digest the food and, as all organisms on Earth do, produce gas. The gas being produced would now be radioactive because the food was radioactive. And so we concocted a food drink made of very simple organic compounds labeled with carbon-14. Now, we didn't just go to the grocery store and pull these organic compounds off the shelf. We used two approaches. One was theoretical and the other was empirical. On the theoretical basis, we looked for materials that had been formed in experiments such as the Miller-Urey experiment, which produced amino acids, carbohydrates, and other building blocks of life, such that it was presumed that early Earth, which had a primitive atmosphere, which was the atmosphere that Miller and Urey exposed to ultraviolet light or electric spark and produced these organic compounds, would have been the method by which life got its start, from the compounds produced abiotically, which in turn would somehow be complexed into the building blocks of life. So we used only compounds that had been formed by Miller-Urey type reactions. But secondly, we tested those compounds on terrestrial organisms. Just as Chris, we roved the planet and went to all of those exotic places or received samples from them, tested thousands of soil samples, thousands of unicellular cultures, pure cultures, wild cultures, against these compounds. And in every case, received responses to tell us there was something alive. If I can have the first view graph, please. Oh, that tells us that we're here. Next view graph. This is how the instrument worked. You saw the arm that went out. It put some soil into a distributor, and about half a cc of soil was plopped into one of 4 rotating chambers. A tiny squirt of nutrient was injected onto the soil. Were anything alive or anything generating radioactive gas, the gas would come up, go through this swan tube, the same kind of tube Pasteur used, that's his link with Mars, and go into a chamber where the radioactivity could be detected. The radioactivity would be monitored through time as it evolved from the chamber. And the beauty of this was that you would use a known amount of radioactivity but never have to worry about counting that or subtracting that from your background because there was a phase change. The radioactive material would have to leave the chamber where it was deposited, go through the Swan's tube where any particulates would be removed and only gas would come through, so you don't have to worry about the background of the nutrient that you originally injected. After all of the experiments were done on Earth and the system was bolted up and Viking was launched to Mars, at that time we did our final test. It may sound a little in arrears, but the schedule demanded it. This was the only time we had a full Viking labeled release flight type instrument. It was taken to Ames Research Center, put into a Mars tank, and some California soil was put into the labeled release chamber. The whole experiment and the chamber was then pumped down to Mars conditions, to Mars experimental conditions. The Mars atmosphere, the temperature 10 degrees C at which this instrument would be maintained through its experiment. This pumping went on for 3 days, and then the experiment was initiated, and the following resulted. Backwards. The instant that the nutrient hit the soil that had been pumped down of water and held at a low temperature for 3 days, we see the curve arising, showing radioactive gas coming out to the tune of about 10,000 counts per minute. Now, we all knew that there's a probability that what we find on Mars is some chemical that will react with the nutrient and produce a curve like that. So the whole biology team debated for years how to differentiate between chemistry and biology. What they came up with was, well, what you do is you heat the sample or a portion of the same sample to a temperature that we think would kill any self-respecting Mars microorganism, that would certainly kill a terrestrial microorganism. You let it cool, you run the experiment again. That temperature is probably too low to damage chemicals that would produce this kind of activity, but ought to show a pronounced effect in killing or damaging the microorganisms. So we did that at California. After we got the test sequence, the active sequence, we heated the control to 160 degrees centigrade for 3 hours, what was selected by the biology team as standard for all the biology experiments as controls. Let it cool, shot the thing again, and you see what we got, zilch, nothing. So that confirmed to all of us that indeed we had a biological sample. And this is what we found in the samples we ran from all around the world. We had never had a false positive in all of the years of preliminary work with this experiment. It went to Mars with a very good pedigree. And what happened on Mars? May we see the next slide. I think the South Pole is down. What happened was very similar to what you saw. As soon as we obtained a sample and injected cycle 1, we had an active curve very reminiscent of the curve that we observed in California in the flight type instrument. So we immediately said, okay, let's go for cycle 2. Let's heat to 160 degrees and run the control. We did, and we got essentially zilch, an excellent demonstration which any place on Earth would absolutely be accepted as positive proof of living microorganisms. We then went to another location, took another sample, cycle 3, and you see the active. At this site, I'm going out of the chronology, but let me explain, we then went into winter, and we could not readily get another sample because we didn't want to repeat the control. We didn't want to waste another chamber. What we did instead was attempt to get another sample. But because of winter's onset, we were told the engineers did not want to risk the sampling mechanism. So we took some of the same sample that gave the active 3 response that had been sitting in the box at 10 degrees C for over 2 months, and we decided we better go ahead and run this thing. Who knows how long the spacecraft will last. We might not make it to spring. And we ran it, and were startled to find no response. So whatever was in that box that we had picked up from the surface of Mars could not withstand sitting around at 10 degrees for a couple of months. That sort of makes a chemical explanation a lot more difficult. 4,000 miles away, we ran a set of experiments on landing site 2 and got essentially the same results. Cycle 1 was the active, and there you see a very strong 15,000 count per minute response. Again, we did not want to go for the 160 degrees C. We're still arguing, can we get a better discrimination between possible biology and chemistry? So we decided, let's have a conference. We got the chemists and the biologists together at JPL, and we said, what temperature would you guys say would discriminate between chemistry and biology in this experiment? And we came up with 50 degrees C. We asked the engineers if they could give us a 50 degrees C heating, and they said they could, and they did. And this was what we got at cycle 2, a very strange response indicating that there's something wrong. So we asked the engineers to check the instrument. They did. They assured us there was nothing wrong with the instrument, but that is a peculiar response. And what we did following that was to move a sample, to move a rock and get a sample from under the rock, because we began to hear, in addition to chemical reasons that may have produced the response we got, there could be physical reasons, such as ultraviolet light hitting the surface of Mars and creating disjunctions or physical defects in the surface material that would react with our nutrient and produce a false positive. So we went out just before dawn, moved the rock, scooped up a sample, put it into the instrument, and there you see under the rock we got a perfectly good response, somewhat less than cycle 1, but still a strong positive response, which again on Earth would be conclusive evidence. Now we decided, let's try again the 50-degree control. And we did. And we shot it through, and we did get a response. This time it was a 70% diminution of the stronger active. But when we examined the data, we found this curve had only, this sample had only been elevated to 46 degrees. So you could make a theory that perhaps the organisms were damaged but not killed at 50 degrees, and at 46 degrees they managed to continue metabolizing. But that really is speculation, and I'm more of an empiricist than a speculator, so I'd like to not stress that point. I said that we knew we would find organics on Mars, and it was a question of which ones. What was the reason behind our feeling? Can we see the next one, please? We know, as I mentioned, that the Miller-Urey type reaction does produce organic compounds. And it was generally thought, as you heard earlier, that the primitive Mars atmosphere was similar to the primitive Earth atmosphere, and therefore it was a foregone conclusion that Miller-Urey type products would be formed. We had even a more dramatic example. In creating one of the life detection experiments, which looked for the formation, looked for the incorporation of radioactive CO2 into microorganisms, looking for photosynthesis, the experimenters found in their preliminary work that when sunlight shines on Martian atmosphere, organic material is produced. Not only is it produced, but even in the face of the persisting ultraviolet portion of the spectrum, it accumulates. So here we have the strange phenomenon that the ultraviolet light is not destroying the organic material, and just shining the sun on Mars should be producing organic materials. And these experimenters said that on Mars, the amount of organics produced over geological time, shouldn't have used the word geological, about, I guess, Marsological time, but they said geological time could be significant. Indeed, it could. We know that there's an influx of organic materials from the early barrage of meteorites and comets and the continuing fallings of these bodies which bring organic compounds to Earth. These objects have been found, analyzed, they have organic compounds. The same thing is happening on Mars. The craters that you saw, some certainly were made by objects containing organic material. Where is it? More recently, in the journals, you will see there's a class of meteorites called the SNC meteorites, which by isotopic analysis and chemical analysis have been generally accepted as coming from Mars. They were ejected from Mars by some of these meteorites, which you saw making the craters on Mars, and they sprayed out with energies taking them out of Mars in a variety of directions, some of which were directed towards Earth, were captured by the Earth's gravity field and deposited on Earth with organic materials intact. These SNCC meteorites all have organic materials in them and are identified as having come from Mars. So here we have organic material that we believe came from Mars that would certainly say there ought to be organic material on Mars. And finally, we have these tiny dust particles, interplanetary dust particles, that are bombarding Earth with some thousands of tons per year, and they have been identified as containing complex aromatic organic compounds, some of them with mass units as high as 670, and the same thing must be happening, although to a somewhat lesser extent because of the lesser gravity, on Mars. So all those reasons say, yes, there are organics on Mars, and indeed the experimenter of the instrument that went to identify those organics, the GCMS instrument, gas chromatogram mass spectrometer, said that yes, there's good reason to identify those organics because they must be on Mars. The way this was to be done was just as is the case with the LR experiment. That same arm scooped up some soil, put it into a tiny oven, very tiny oven, 600 milligram content. And the oven was then turned on. If there were any organics present, they were evaporated. They went through a gas chromatographic column to separate them into discrete segments, which then were fed into a mass spectrometer for identification. The instrument was a marvelous attempt at miniaturizing something which on Earth in our laboratory is about almost 5 times as big as this whole podium. And that instrument was placed into something far less than a cubic foot. And you had to give up certain things to do that, amongst which are sensitivities and the ability to look in particular areas and discriminate. The GCMS data were eagerly awaited on Mars because obviously they would have great importance with respect to interpreting the LR data. And the next view graph shows we all waited and absolutely no organics were detected as indigenous to Mars. Some organics were detected which were a contaminant in the instrument from Earth from its cleaning process, but nothing that could be attributed to Mars. The only compounds detected were carbon dioxide and water. And because of the mode in which the instrument was operated, the experimenter said that the values for water at the VO1, at the number 1 landing site, are very less precise than at the number 2 landing site, and the mode in which it was operated at Lander 1 prohibited determining carbon dioxide. So the only results pertinent are those at Viking Site Lander 2 with respect to water and carbon dioxide, the only compounds detected. Moreover, these analyses were run at different temperatures, 200 degrees C, 350, 500. Then the same sample under the rock where we got a response was run 50 degrees, 200, 350, 500 in an attempt to see how the increasing temperature drove out the volatiles. Now the problem was to do that, days had to separate each heating. So it wasn't a continuous heating. You would heat and then you'd wait, let the thing sit around, and then heat it again. What happened then was a theory was proposed as the peroxide theory to explain the LR and the GCMS results. The theory is that in the upper atmosphere of Mars, the sunlight disrupts water molecules into hydrogen and hydroxyl ions. 2 hydroxyl ions unite to become hydrogen peroxide. The hydrogen peroxide falls down to the surface of Mars, where it does two things. It gets scooped up in the material that we put into the LR, and the hydrogen peroxide reacts with our nutrient to give us a false positive. And also, the hydrogen peroxide consumes all of the organics, which explains why the GCMS found no organics. The sensitivities of the two instruments, however, are instructive. In order to detect organic matter, the GCMS requires the organic matter equivalent to that provided by 1 million bacteria. It doesn't matter whether they're living or dead, unless, like Chris's fossils, the dead ones might have lost a lot of their organic matter. They've got to still have the organic matter. You need the organic matter equivalent to 1 million bacteria. The LR finds 10 bacteria and doesn't require any growth. That's the whole rationale behind its extraordinary sensitivity. When we were looking at the record prior to the launch, NASA sent all of the experimenters a variety of samples to test, as I mentioned. There was a sample from the Antarctic taken from high in the dry valleys that Chris mentioned that the GCMS examined and reported that it failed to detect any organic matter. Years later, when I was browsing the literature files, I found that a classic chemical analysis made at the same time by the same team found indeed there was 0.03% organics present in that sample. And I found just last week, this thing never ends, I kept looking and I found that not only was there organic carbon present, but there was 0.003% organic nitrogen, a matter which has never been addressed in discussing these Viking findings. Well, as I delved into the files, I also found that at the same time that the GCMS was unable to detect organic material in Antarctic number 726, we had run 726. I pulled the record out and we had detected there was more radioactive material in the nutrient than came out. The radioactivity which evolved was more than the radioactivity contained, however, in one nutrient. So more than one nutrient was involved. We can't tell, however, whether that was limiting or whether the oxidant was. And there's, unless we measure the oxidant some way, perhaps by MOX, we can't really make an educated guess on that.
Yes. What about the possibility of flying a microscope to directly observe the samples while they're active?
Flying?
A microscope to look at what is going on to see if you can directly observe microorganisms in a sample that is...
A lot of effort was put in the Viking development phase into an automated microscope. Trouble with the microscope is it doesn't really tell you very much. If you see a small spheroid, is it alive or not? You can't tell. And some people played games with moon sample dust, taking photomicrographs and saying, hey, is this alive or dead? And you really can't tell. Especially if you're going to look at an alien life, you wouldn't know what you were looking for anyway. So it'd be very difficult. Yes, Craig?
Dr. Levin, could you elaborate more on the poison you talked about? I know you talked about the pH and the endothermic or exothermic heat of reaction, but the poison would be an inert material which would not chemically react, so you could discount chemical reaction. Could you elaborate more on that so that we could determine once and for all whether that was life that you saw? What type of poison would you use? Would it be cyanide, or what type of poison are we talking about?
In the work, our early work with our experiment, we would take it out to, say, Death Valley. And at that time, it was called the sticky string experiment. It fell out on the sand, and a little bit of sand granules were pulled in, and then the nutrient was applied. We did not use heat as our control. In those days, we used an antimetabolite. We used an alcohol-based material. And this worked very, very well and would be less likely to be susceptible to confusion, I believe. But I think we ought to do both in a future experiment.
Dr. Levin, we know that on Earth there are anaerobic microorganisms that do consider life to be a terrible poison, and that was in fact the dominant life form probably on Earth in the early stages. Is there any analogous organism that would actually expel oxygen or create and store oxygen as an oxidant on Earth that you know of?
That's where our oxygen comes from. Green plants, algae primarily in the oceans, take in the CO2 and produce the oxygen. And on Mars, there is an unbalance, which is what gave rise to the oxidant theory, the hydrogen peroxide theory. There's CO2 in predominance on Mars, and there should not be. As Chris pointed out, it ought to be precipitated out as carbonate, but somehow it's being recycled. So in order to recycle or explain how the CO2 remains in the Martian atmosphere, hydrogen peroxide was conceived of as then reacting with the CO2, with the carbonate and reliberating CO2. But the other thing that could do that would be living organisms.
Okay, thank you. That's a very good example. I'm sorry, I didn't think of that.
Yeah, Dr. Levin, something I've wondered about for years, and maybe this isn't possible, but has anybody ever checked the Mars rocks that have fallen in the Antarctic for living organisms?
The first person to try that was Louis Pasteur. He got hold of the Orgueil meteorite, which fell near Paris, and he had just developed this technique for making hay infusions and brewing up cultures of organisms in there. So he actually split open a piece of that meteorite, scraped out some particles, put it in one of his hay infusions, got nothing. I don't know whether anyone has attempted that with the meteorites today, the SNCC meteorites. I do know they spent a lot of time analyzing for organic matter. Chris, do you know anything about that?
You can't separate out any extraterrestrial life because there's been too much terrestrial contamination, but Gil is correct that they have been able to isolate what they think is non-terrestrial organic matter, and hence they think that this is organic matter from Mars in the SNC meteorites. And interestingly enough, its carbon-13 to carbon-12 ratio is -20 per mil, which is the same ratio you find in living systems on Earth, but not in carbonate, which further makes the GCMS results mysterious.
The whole thing is very tantalizing, really is. Thank you very much.
Thank you, Gil.
Thanks.
Testing.